esophageal cancer. It's a diagnosis that carries a really heavy weight, and statistically, it's still one of the most lethal cancers out there.
It really is, and today we're focusing in on one specific type, esophageal squamous cell carcinoma, or ESCC.
Which is, I mean, it's something like 90% of all cases worldwide. It's the dominant form.
Exactly. And a big reason for that lethality is that our current treatments are, well, they're just not specific enough.
You're talking about things like chemo, surgery, some of the broader immunotherapies.
Right. They're broad spectrum tools. Think of it like using a flamethrower to get rid of a few weeds. It might work, but the side effects for the patient are just devastating: fatigue, nausea, bone marrow suppression. The cost is huge.
So the whole field is just crying out for more precision, and that's what makes this deep dive so exciting. We're going to walk you through a completely new strategy, one that targets a fundamental physical process in the cell.
It's called liquid phase separation, LLPS. Sounds like physics, but it is pure critical biology.
And at the very heart of this new approach is one specific protein, a transcription factor called AP2 beta. Or TFAP2 beta.
And that's the key player. Researchers found that this protein is a powerful tumor suppressor.
But, and this is the whole story, it can only do its job if it can form these little liquid droplets inside the cell nucleus.
So our mission today is to unpack that entire scientific journey for you. How do they even find this protein? How does this weird liquid droplet mechanism actually work?
And then the payoff. How do they design a new drug, compound A6, that actually uses this mechanism to fight the cancer with incredible precision? It's a story that starts, with a very practical problem.
Just getting good samples.
Okay, so let's jump into that first big hurdle, the technical breakthrough. The main problem with setting early-stage ESCC is the samples themselves. They're tiny.
They're minuscule, and they're hard to preserve. So traditional methods, especially for deep genomic analysis, they just fail. You don't have enough starting material.
And this is a huge deal, right? The main technique you want to use here is something called ATAC-seq.
It is. Assay for transposase accessible chromatin using sequencing. It's essential.
And what did that actually tell you?
Imagine the cell's DNA is a giant library with millions of books. ATAC-seq tells you which bookshelves are currently open and accessible, which genes are ready to be read by proteins like that TFP2 beta.
So without that map, you're flying blind. You can't see the cancer strategy, but you can't run the assay if you only have a trace amount of tissue.
Exactly. So what these researchers did was they completely optimized the protocol. They moved away from harsh methods and developed a gentler, two-pronged approach.
What did that involve?
It involved using mechanical digestion combined with two specific enzymes, collagenase and dispase. These enzymes can very carefully break down the tissue matrix without shredding the precious chromatin inside.
And this wasn't just a small improvement.
No, it was a game changer. Their success rate for generating high-quality data from these tiny samples shot up to over 80%. That just blew the door wide open for this kind of research.
So with that working, they could finally build a real data set.
A fantastic one. They collected 66 paired samples from 28 patients, and they were able to separate them into three groups. The healthy tissue nearby, which they called paracancerous, or P.
Okay.
Then the tumor that was just invading the first layer, the mucosal invading tumor, or TIM. And then the more advanced submucosal invading tumor, TINIS.
So you have a clear progression: healthy, early cancer, and more advanced cancer.
And they combine the ATAC-seq data, the accessibility map, with RNA-seq, which tells you which genes are actually being turned on or off.
Putting those two data sets together must have given them a huge amount of insight.
It did, and they zeroed in on the transcription factors that were being downregulated, the defensive players that the cancer was managing to shut off.
And three names popped out.
Three big ones: TFEP2 beta, another one called NFEX, and a third called ID4.
But TFP2 beta was the prime suspect. Why?
Because its expression levels just plummeted in lockstep with the cancer's progression. It was high in the healthy tissue, lower in the TIM stage, and even lower in the TIMism stage.
A really clear correlation.
A perfect correlation. And then they checked it against the huge Cancer Genome Atlas database, the TCGA, and that data confirmed it. Patients with low TFAP2 beta had a much, much worse prognosis.
So all signs point to this being a major tumor suppressor.
No doubt. And they confirmed it in the lab. When they forced cancer cells to produce more TFA2 beta, Two things happened. The cells started dying off through apoptosis.
Programmed cell death.
Right. And they also stopped multiplying, they stopped moving, they stopped invading. They confirmed all of this in 2 different mouse models as well. It's a key defensive protein.
Okay, let's just pause there for a quick recap.
Sure.
So the first section, we've basically found our target. Researchers solved a massive sample sourcing problem with a new ATAC-seq method that allowed them to screen early-stage cancer with incredible precision, and they found that a protein, TFA2 beta, was being systematically shut down as the cancer got worse.
And now we get to the real question. How does this protein actually work? What is it about it that stops cancer?
Which brings us to phase separation, LLPS. This is probably the coolest part of the whole story. I mean, we always use the oil and water analogy for this.
It's the best one. LLPS is how cells create compartments without membranes. They're these tiny, dynamic droplets, membraneless organelles that act like little concentration hubs or microfactories.
They just gather up all the necessary tools for a specific job in one place.
Exactly. They pull in specific proteins and RNA to really high concentrations, and that just massively speeds up whatever reaction needs to happen.
So when TFPT beta is doing its job correctly, it should be forming these little factories.
And that's precisely what they saw. In healthy cells, TFAPT beta forms these little droplet-like spots, or puncta, in the nucleus. But in the cancer's tissue, they're almost completely gone. The cancer dissolves them.
Okay, but how do you prove they're actually dynamic liquids and not just, useless clumps of protein?
They ran the classic tests. First one is called FRAP, fluorescence recovery after photobleaching.
Right, you use a laser to bleach a spot on the droplet and see if the color comes back.
Exactly. And it came roaring back in seconds. That speed tells you molecules are constantly moving in and out. It's fluid, it's dynamic.
It's not a solid speck. And they also showed it was dependent on electrical charge, which is a hallmark of LLPS.
Yes, this was the smoking gun. They shifted the environment to a high alkaline pH of 10, and the droplets just dissolved instantly.
Wow.
But the moment they brought it back to a normal pH of 7, poof, the droplets reformed. That proves the whole process is driven by electrostatic interactions, positive and negative charges pulling the molecules together.
So what part of the protein is actually responsible for this?
It comes down to these long, floppy, unstructured parts of the protein called intrinsically disordered regions, or IDRs. TFAPD is 2, but all the action is in a small segment called IDR2.
IDR2 is the command center.
It is. They proved it by deleting it. No IDR2, no droplets. Then they got even more specific. They took just 6 positively charged amino acids within IDR2 and mutated them to be negatively charged.
Flip the switch.
Flip the switch. And condensation was completely abolished in the test tube and in living cells.
And here's the absolute killer link, the connection between the structure and the function.
This is the cornerstone of the whole paper. Those mutants that couldn't form condensates, they completely lost their ability to suppress tumors. No more apoptosis, no more stopping proliferation.
They were duds.
Completely. But then they did the reverse experiment. They took that broken mutant and they artificially gave it the ability to phase separate again by fusing it to a known LLPS domain from another protein.
And it worked.
It worked perfectly. It restored the full anti-tumor activity of TFAP2 beta.
So that's non-negotiable proof. The physical act of forming that liquid droplet is absolutely necessary for the protein to work.
It's not just about the protein being there. It's about its ability to cluster and form that microfactory.
What is that factory actually doing? What's its job?
To figure that out, they threw 5 huge data sets at the problem, looking for genes that were directly controlled by the healthy TFAP beta cell, but not by the broken mutants. And they found only one common direct target.
Just one.
One, a gene called zinc finger protein 131, or ZNF131.
And I'm guessing ZNF131 is not a good guy here.
Not at all. It's a potent oncogene. It promotes cancer, and the job of the TFE2 beta condensate is to shut it down.
Ow.
It's a physical mechanism. Think of it like a turf war. Normally, a different protein, an activator called YAP1, binds to the ZNF131 gene to turn it on.
Okay.
But when the TFAPD beta condensate forms, this big, dense liquid droplet physically sits on that same spot on the DNA. It blocks YAP1 from getting in. It's not a chemical reaction, it's a physical blockade.
Wow. So it just muscles the activator out of the way.
Exactly. But it has a second job, too. The droplet also acts as a platform. It recruits those other 2 tumor suppressors we mentioned earlier, NFAX and ID4.
So it brings the whole defensive team together.
It does. They showed that NFX and ID4 co-condense with TFA-beta, and being pulled into that high concentration environment actually boosts their own activity. It helps NFX bind more strongly to its own DNA targets, for example.
Okay, time for our second recap. This is where it gets really, really interesting. So TFA-beta forms these liquid condensates driven by a specific region called IDR2 and its positive charges.
Right.
And this little factory has a two-part mission. First, it physically blocks a cancer-promoting gene, ZNF131, by keeping its activator away.
And second, it acts as a recruited platform to bring in other tumor suppressors like NFX and ID4 and make them more effective.
So now we know exactly what's broken in the cancer. The next logical step is, how do you fix it?
Precisely. If the cancer strategy is to dissolve this condensate, the therapeutic strategy has to be finding a drug that enhances it, that stabilizes it.
Which is a huge challenge because there basically weren't any LLPS targeted drugs for ESCC.
None. So they had to go hunting. They used a really sophisticated virtual screening process, using computers to analyze thousands of compounds to see which ones might interact with TFA2-beta.
And that search led them to one specific molecule.
It led them to compound A6. A6 was the strongest inhibitor of ESCC cell growth they found. And the reason was that it did something completely new. It actually enhanced the formation of the TFA2-beta condensates.
It rebuilds the very thing the cancer is trying to tear down.
Yeah.
How does it do that? Where does it bind?
This is the really elegant part. It doesn't bind to that floppy IDR2 region. It binds to a specific rigid pocket on a totally different part of the protein, near the DNA binding domain. It's an allosteric regulator.
So it's not directly pushing the molecules together. It's changing the protein's shape from a distance.
That's it, exactly. And they found the precise keyhole it fits into. 2 amino acids, arginine 382 and asparagine 380.
And they proved that's the spot.
They did. They mutated those 2 residues, and A6 couldn't bind anymore. And when it couldn't bind, it lost all of its anti-tumor effects. It couldn't promote condensation.
So the mechanism is it's structural genius. A6 docks into this rigid pocket. That binding causes a subtle long-range change in the protein's shape, which then exposes those critical positive charges on the IDR2 domain.
You've got it. It's like turning up the power on a set of magnets. It just boosts the natural attraction between the TFA2-beta molecules, making the condensates stronger and more stable.
And the efficacy data on this must have been pretty compelling.
It was beautiful. A6 inhibited proliferation, migration, invasion, all in a clear, dose-dependent manner. And crucially, it showed great specificity.
Meaning it hit the cancer cells harder than the healthy ones.
Much harder. Normal esophageal cells needed a significantly higher dose of A6 to be affected, which suggests a really promising therapeutic window.
And they didn't just keep this in a petri dish. They moved to more complex models.
3 of them. First, patient-derived organoids. These are mini tumors grown in a lab from actual patient tissue. A6 shrank them, and visibly you could see the TFP2-beta droplets reforming inside the cells.
Incredible.
Second, in patient-derived xenograft mice, where you implant a piece of a human tumor, A6 significantly inhibited tumor growth over 7 weeks.
And the third.
A standard subcutaneous xenograft mouse model. Same result, strong inhibition of tumor growth.
And maybe most importantly for any new drug, what about toxicity? What about side effects?
This was the best part. Across all the mouse models, there was no impact on body weight, no signs of toxicity to any of the major organs, heart, liver, lungs, kidneys, nothing.
That is the gold standard. High efficacy against the tumor, minimal damage to the patient.
So to pull this all together, this research really establishes TFA2-beta and its ability to phase separate as a critical and, more importantly, an actionable mechanism in this cancer.
And it validates compound A6 as the very first-in-class LLPS-inducing drug for ESCC. A drug that works by enhancing a natural defense, not by poisoning something.
That's the key takeaway. Now, the researchers are very clear there's still a long way to go.
Of course. This is still early days.
They need to do all the hard work on pharmacokinetics, how the body absorbs and processes the drug and long-term safety studies.
And there's still some deeper mechanistic questions too, right?
Oh, absolutely. Like, does this droplet physically reorganize the whole neighborhood of chromatin around it? And do those other proteins, NFIX and ID4, form their own little condensates, or are they totally dependent on TFAP2-beta-Cs?
But that question about reorganizing the neighborhood is just fascinating. So let's leave our listeners with a final thought, one that really captures the paradigm shift we've been talking about.
Go for it.
We've just learned about a tumor suppressor that works by forming these liquid condensates, and a drug that fights cancer by deliberately making those condensates stronger. So here's the question. Do you think the future of cancer therapy lies less in our traditional playbook of finding an enzyme and inhibiting it, and more in designing molecules that deliberately enhance or stabilize the beneficial phase separation of other key tumor suppressors across all types of cancer? It suggests that maybe stabilization, not inhibition, is the next great therapeutic frontier.